Yeah, what's up?
Hello.
Hi.
You're Dr. Jacob Wirth.
That's true.
I'm not Dr. Jacob Wirth.
Also true.
Do you want to hear my catchphrase that I came up with?
Did you just come up with it just now?
No, I came up with it a couple days ago, and I've been waiting. I'm really excited about it. I put weeks of work into it.
If you came up with it just a couple days ago, how did you put weeks of work into it?
Time travel.
All right. Yeah, what is it?
Let's get lay.
Because layman.
Because layman.
Right.
Do you want to introduce the paper?
So the paper is a Nature Communications article. So it's kind of a smaller, not full beefy paper. But Nature is obviously one of the big journals.
There's not going to be any beef?
Not this one. It's mice and monkeys.
Of mice and monkeys.
Of mice and monkeys. That should have been the title, but they really dropped the ball. The paper is titled, Triptonide is a reversible non-hormonal male contraceptive agent in mice and non-human primates. They mention mice and monkeys, but...
Yeah, I think I'm going to retitle this Of Mice and Monkeys.
You want a big pen?
Yeah.
So I'm not a developmental biologist by any means.
What are you?
I'm a protein biochemist by training. And I guess right now I'm working in pharmaceutical development and enzyme engineering. So...
And is this your assistant who just crawled up onto you?
He is now. He's also not Dr. Jacob Wirth.
None of us are. None of us on this couch are. Except you, of course.
Except me.
Dr. Jacob Wirth.
You were 1/3 wrong.
Well, let's get lay.
Let's get lay. So half of this paper, I am pretty well versed in what they did. And half of this paper, I'm kind of knowledgeable and take some of what they said on faith. But if you've ever read a Nature Communications article...
I haven't.
Most people haven't. They glance through it because there's like 20 supplemental figures and 4 figures in the paper. It's the kind of paper that you hide a bunch of stuff in the parts that you don't want people to read. It happens.
Okay. So take me through it. What is this about? You mentioned it's about guys' balls?
Yes. It is about a potential non-hormonal male contraceptive. So we don't have the pill for men. And you can say that there are any levels of decades in the history of politics or sexism in the pharmaceutical industry says we don't have the pill for men. But from a science standpoint, it's difficult. It's difficult and it could be dangerous, and you can have a lot of things go haywire. And when you develop a pharmaceutical, you also have to remember that it's people, so you need compliance. So if you have people become really, really ill, it doesn't matter that it doesn't kill them. They might just stop taking it. So if you're actually developing an oral contraceptive, you have to make it so people will take it, not just make it work. So Triptonide is a natural product from the Tripterigium wilfordi plant. It's a herb. It's a Chinese herb. It's been used for thousands of years.
It's a herb, you say?
It's a herb.
Oh, I've never heard you say herb before.
Probably because I haven't.
That's beautiful. I love that it's coming
Anyway,
out now. Can you do the rest of this in a British accent?
It's a Chinese herb. I can't. You only get the one word. That people have been using for as like a medicinal plant for decades and centuries and millennia. And it's been noted that people who are taking it regularly, men are infertile. So the whole basis of this paper and this study that was years long was clearly something's in this medicinal plant that leads to male infertility. People don't know what it is. There are a couple natural products from the plant that people already use for various things. Triptonide is a chemotherapy agent. They found Triptonide, which is a slightly different molecule.
Super slightly?
Super slightly. Like the difference between like one hydrogen. One has an OH group, an alcohol, and one has just an O ketone. So instead of having a double bond, it has a bond going to an H. So it's literally one atom difference. And one of them is a very toxic chemotherapy agent. And one of them is apparently a non-hormonal male contraceptive.
Oh.
Yeah, it's cool, isn't it?
Science.
Science indeed. I want my tea, but if I move, the magical will stop.
You don't get your tea. You have a cat instead.
Yeah, I guess it's a good trade. So they synthetically made this because they tried to purify it from the plant, and it had a bunch of the toxic parts left over. So they couldn't do that. So they had to make it synthetically, which is harder and more expensive and time-consuming and probably dangerous. I don't know how they made it.
Is it easy to know which parts are toxic, or did they find that out by killing off mice and monkeys?
I think they knew about Triptonide and its toxicity already. So they saw that when they purified the Triptonide, they saw a bunch of Triptonide left over, and they're like, we can't.
Oh, so it was the same.
Yeah. They're like, we can't give this because we already know that it's going to be toxic. We're not looking to make a toxic male contraceptive. We already have those.
Do we?
This isn't the first oral male contraceptive that they've tried, and a lot of them end up being toxic or cause like hormonal issues or psychological issues or something. It's bad.
Well, I would put forth that there are many hormonal and psychological issues that come forth from the female pill that is widely used.
That would generally be the argument that is put forth when something like this is mentioned. I try to stay away from that because the politics surrounding a different pharmaceutical has nothing to do with this pharmaceutical.
Fair, I guess.
The female pill is not on trial here. The data surrounding this male pill candidate is on trial. And that's what they say. And they don't mention the female contraceptive at all, except in the discussion section when they're like, compliance is a big deal, so we have to make sure the pill doesn't suck. Otherwise, people don't want to comply. We've already seen this in other types of contraceptives. Because, I mean, like, condoms are pretty good, but there are reasons X, Y, and Z that people might not want to use them, so they don't. So the study, essentially what they do here, I mean, unless you had any other questions going.
Oh, just walk me through it.
Yeah.
I don't know where we are. I am not a guide in science land. You hold my hand and you drag me through this horrible, rough terrain.
Mm-hmm. The very first thing that they mention is that there is a female, the pill. There's a shot that men can get to make them infertile, and then it takes a shot to reverse it. That's 2 shots. And taking a pill is a lot easier than that. It doesn't require shots. So there's reasons that you would go down this road for this study anyway, even though we have things that already do this. We don't have the perfect storm of something helpful.
That shot thing is fucking fascinating to me, and I had never heard of it, and I would like to know more some other time.
Yeah, I don't know much about it. I've never really looked into it because I've never been interested in getting infertility shot. But yeah, it's essentially a contraceptive of a different route.
It just shuts everything down
guess.
Until you unshot it.
Mm-hmm.
That's so strange.
I don't know what what's about it is reversible, but I know it's not reversible on its own.
Okay.
I'd have to look up more about that. But those are the kinds of things that they mention. So they go, okay, well, we don't want something super that messes with your hormones because some people just can't take that and it's painful or it screws up your body or whatever. Basically, like you said, a hormonal, the pill is a downside. It would be cool if we didn't have that downside. They wanted a male contraceptive, so they thought spermiogenesis, the making of the sperm. They wanted to target that. And they wanted to target it in a non-hormonal way because you can add a lot of hormones that make it so somebody can't make their birthing cells.
I know for a fact that spermiogenesis is a JRPG main villain.
I would be terrified.
That you don't know is the villain until the very end. You think it's someone else.
The villain behind
spermiogenesis.
No, it was sperm all along.
You know, it usually ends up being sperm all along.
It's true. I've been to Japan, so I know. But they wanted it to be non-hormonal because that is easier to make it acceptable as far as side effects go. So they try to go for one of these natural products from this herb that I already butchered the name of, don't need to say it again.
Okay.
And they go with Triptonide. So the first thing they do is they introduce it to mice. They do a mouse study because that's what you do, because no one minds if you kill dozens of mice in your study. But if you jump straight to humans, that would be terrible.
In your study.
In your study.
Yeah.
Yeah, you have to go through the review board. You're not supposed to just kill mice.
You aren't?
Unless it's for science. But other people have to decide it's for science.
What if you've named your evil overlord science and you kill for science?
I feel like that's a question for the ethics committee. I would say that's an acceptable loophole.
Perfect.
They gave the mice the drug and they saw that basically nothing bad happened to the mice other than their sperm started having bent backwards heads. And I don't remember the name of the exact phenotype they say, but it's basically like head bent backward phenotype.
All right.
And you look at them under the microscope and they have in their first figure a lot of charts that basically have a bunch of horizontal lines saying nothing changed. Look at all of these different things that didn't change, except this picture of the sperm with its head bent backwards. So that's really important because if testes size, sperm count, like if all of these things don't change at all, it means the other like hormonal system in the mouse is basically fine. Other than the sperm is being deformed.
With head bent backward syndrome of your sperms, can it still fertilize? Or
No.
That's it.
They can't even, so they actually do the experiment where not only when the mice are bred, but with in vitro fertilization, they took the sperm and mouse eggs and tried to do actual like test tube fertilization, and they still couldn't get it to work. The sperm doesn't go into egg properly because the head is malformed.
That's amazing.
It's awesome.
Once they made the thing not toxic, it immediately worked in the way they wanted it to.
Yeah.
That seems very lucky.
It's incredibly lucky, and they mention at different points in the study, like, it's clear that there is this one part of the sperm morphology that is gated by one protein and can be disrupted with one interaction, and it causes this very specific type of thing to go wrong with the shape. And that shape happens to make it so they can't go into eggs. And in general, can't swim at all. They can't direct themselves because they can't cut through the cytoplasm properly. And you actually see the inner cell juice, the cytoplasm. You can find remnants in the little hook that the sperm makes. So it's like, it doesn't make it and spit it out properly. It like gets stuck and jammed. It's weird. They have a couple images, and I don't really know what I'm looking at because I haven't seen a lot of sperm under microscopes.
This is like when pregnant women show the ultrasound.
Yeah.
I've never been able to see it.
There's
no baby in there.
Like, they put the control in next to the treated sperm. Those don't look different. But they're different.
Sperm conspiracy.
And then some of the pictures, you can see that the sperm is kind of deformed, but I wouldn't have, like, guessed that was deformed. It could totally be that's what sperm look like. I don't know. But that's why they're developmental biologists, and I'm not. The big part of this...
That is why. As a child, they could recognize different sperms and know exactly what it meant, and so they had to go into developmental biology.
It was their calling. The big part of figure 1 is just look at all these horizontal lines. Like, these are the same, these are the same, these are the same, that's the same. Like, all of these things, it's just horizontal is the point. When you're treated, none of these numbers go up or down. And that's cool, because you don't want anything to go up or down unless you meant for it to go up or down.
What are these numbers?
So we have testes weight, sperm count, testosterone levels in the testes, testosterone levels in the blood, FSH, LH, like growth hormones, and various things. Body weight doesn't really change.
Does it say what the general testes weight is? Because
for a mouse, it seems to be they don't have a number.
Oh.
They don't have a unit. They have a number. It's
one mouse testes unit.
Oh, it's testes weight times 1,000 over body weight. So 1/4,000th of their weight.
Oh, okay.
And I don't know what a mouse weighs.
Well, that's not a neat little fun fact.
No, it's not. Well, I don't know that our testes are 1/4,000th of a human's weight. That seems pretty high, actually.
We don't share testes. Both of us have our own testes.
That's why they don't weigh as much.
Oh.
Because each human has to have their own set.
Oh.
Except this line, and that's sperm with normal morphology percentage. And it goes straight down. So that's the one thing that changes. Sperm shaped weird. So figure 2, they go to monkeys. They say, okay, looks like we have a good handle on what happens with mice. Well, let's go to primates. And they go to cynomolgus monkeys? I don't know anything about that type of monkey. I don't think I've ever heard of that type of monkey. It's just some monkey. I don't know why
Whoa. Racism. Reel it back.
All primates look the same. I don't know why they went to that. I don't know if that's like a good reproductive model as a monkey, because like you hear about bonobos, you hear about chimps, so maybe there's a reason they went to these monkeys. Maybe they're just cheap.
They just live next door. They put out a call.
It might just put a help wanted sign right outside the lab, and all of the cynomolgus monkeys show up.
Yeah, they take the thing like, you need help? I want to help.
I help.
I'm monkey.
We'll do experiments for food. Maybe they have like a short generation time because you're doing reproductive studies. I don't know.
Does it say how large their balls are? Because if they have like the heaviest balls of all monkeys, then it'd be easy.
The easiest to.
Very easy access ball monkeys.
Litter size, motility, sperm deformity. They don't say testes weight
Damn,
in
that's
any.
a cute little monkey.
Yeah, it is. That's figure 3. We're not there yet. In figure 2, they show basically the same stuff. Though in that figure, you can actually see the sperm. You do see it like swirl around. That is a bent backward sperm. This is a not bent backward sperm, and that is a bent backward sperm. And that's the only difference. And that trying to go forward can't bump into an egg. This is the whole paper can come down to this is what it looked like before and this is what it looked like after.
Yeah.
It's pretty neat. But it does, it just makes it look like it's a little swirl.
I have seen contortionists who put all of their body back behind their head though, and then they could still scurry at you.
That's true. But could they enter your eggs?
They play the cards right.
Depends on what they're wearing.
I'm a bit of a tramp.
Do they know that, though?
Everyone knows it.
Yeah. You see kind of the same graphs. Sperm count, however, does go down this time. And like as sperm deformity goes up, sperm count goes down. And they mentioned later in the discussion that that's basically one of the biggest things. I think testes shape and sperm count do get wonky with the monkeys and don't with the mice. And they are not sure why, because based on all of the other evidence and all of the other experiments they do throughout the study over the months and years the study took, everything points to the fact that the Triptonide is affecting the same thing in the same way. So that is a purely developmental pathway issue of monkeys. More than one thing gets turned off by this gate. When you block this protein, monkeys have more things go wrong, and that's why they start losing sperm count. Mice don't have that thing go wrong.
What shape do their balls become?
I think they don't elongate it enough. I'd have to find the exact wording again.
Enough for what?
I don't know. They don't elongate as much so other monkeys don't make fun of them.
Oh.
Yeah. Locker room talk. I see these. I don't know what this says. I don't know what that says. The only graphs that really make sense in this figure is the one that says deformity percentage goes up. I don't even know what does that mean? And then sperm count goes down.
So human people have been taking this herb and becoming infertile.
Yeah. Among the other things that they treat it for. Like, that's not what the herb is for.
Have their balls been mocked ruthlessly?
I feel like you'd have to take it for a long time. And if the difference between, like, monkeys and mice have this deformity issue, who knows what the difference is up to humans? Like, who knows what could
Of
happen.
monkeys and men.
Yeah, of monkeys and men. You never know if men, like, male human testes would have an elongation problem or something.
Are men's balls the monkey or the mice?
That's what clinical trials are for. That should be what the proposal for the clinical trials is. When they go to the FDA and they're like, are the men's testes the monkey or the mice?
Should be the mouse.
Right.
I caught that immediately, and I was just going to try and let it pass, but then you tried to repeat me.
I
cannot let that
should
stand.
know better.
I'm the lay.
So...
You don't get lay. Only I get lay.
Yeah. We went over about the trampiness. You don't have to remind me. So...
I'd be pretty trampy for that baby monkey, though. That's so cute.
Yeah. You really gotta get a picture of this monkey. Anybody just listening is...
Are you licking me? Was that a lick? You don't lick me.
So figure 1 and 2 are you can make mice and monkeys infertile, right? Awesome. Very important for a contraceptive that it's a contraceptive.
Mm-hmm.
You gotta make it reversible, otherwise you're not giving somebody a contraceptive. You're just...
Well, I mean, it depends on what your goals are.
Chemically castrating them.
Yeah, that could be useful.
We already have things that do that. Basically, in figure 3, you see a bunch of charts that have horizontal lines, and then a bunch of charts that have curves. And the curved ones basically say, after a couple weeks, the monkeys go back to normal. And then all the horizontal lines, like litter size, litter interval, mating frequency, all of those are exactly like they were never on it. Like the monkeys that were never on the drug, everything's the same.
So this is just when they stop it?
When they stop it, after a few weeks, the monkeys that have stopped and the monkeys that never started have indistinguishable mating patterns, children.
Ball size?
Probably ball size. There's no ball size chart. Like I said, there's no ball size chart for the monkeys.
Ball shape.
Nah, it doesn't say that either.
Ooh, mocked forever.
Mocked forever. That part never goes back. It only takes like a month, too, before things are back. So were a human taking this, if this scales to human, you'd take the pill and you'd be infertile after a couple weeks. You'd be infertile the whole time. You'd stop taking it and you'd be fertile again after a couple... Are men fertile? You'd be virile? I don't know what the term is. And I don't know...
I like the concept
of a fertile male.
Yeah.
I don't know enough about the pill. Like the pill. I don't know enough about the normal pill. How long after you stop the pill do you... I don't know enough about the pill that when you stop taking it, how long before you can have babies again?
It varies depending on the person and depending on how long you've been taking it.
I imagine, is it a couple weeks? Is it like a week?
I'm sure there are people that can just pop a baby in away.
Aren't there like sitcom plots where, oh no, I missed a day and then now it's this pregnancy scare?
Yeah, we should base our scientific knowledge off of sitcom plots. You're absolutely correct there.
What about when they're science sitcoms?
Those don't exist.
No, they don't.
They tried that one time and it lacked all science and humor.
All science and humor. So the thing's reversible. It works. It works in primates and it's reversible. So these are kind of like the go-tos. If I said you need a male contraceptive in human men, those would be the things that you ask first. Like, does it work? Does it work in primates? Is it reversible?
I think you're thinking small here. Can we get a male contraceptive in human women?
Not without casualties. So here, lay person, if we're gonna, let's get lay.
Oh, no, I get lay.
You get lay.
You don't get lay.
So that was to them.
Yeah.
Okay. If all
let's get lay.
y'all are gonna be the lay people here, Those are the first 3 questions I said. Like, from a scientific standpoint, you're going through your experiment. Does it work? Does it work in primates? Is it reversible? What's your next question?
How does it taste? Is that right?
I don't think they asked the monkeys. No, it's not right.
Is that right?
Of course it's not right. The next question is, how much does it take? Like, you know...
I'd get there, but I want to know how it tastes first. If I'm going to take it.
Because I can give you literally any drug, if you take enough of it, will make it so you can't have babies, because it will have killed you. Oh. So, like...
You know, why aren't we putting money into those studies?
How much...
How can we make a corpse infertile?
I don't think we're supposed to encourage that line of thinking. They look at the dose rate, and they get the minimum effective dose and the EC50, which are different. One is basically the amount that it takes to... The things you want to know is, how much does it take until everything in the study was infertile? How much does it take until, on average, things were like half and half? Because those are the kinds of numbers. It could be a really steep curve where those are basically the same number. It's like a switch. Or it could be really, really big curve in that you have to take a lot before you're sure the entire population is infertile. And that would be shitty, because then you would have your doctor prescribe to you orders of magnitude differences depending on the person. Mm-hmm. That would be annoying. You really want it so that the difference between effective in people and effective in everyone is basically the same. So they get those numbers, and for the monkeys, they get the minimum effective dose is 0.1 milligram per kilogram of body weight. And if that scales, the first thing my colleague Chris said was, if that scales, that means most people could use this, and 100 kilograms is like over 200 pounds. So most people could be rendered infertile by a 10 milligram pill. 10 milligrams is nothing. Think of the last pill that you took. Do you know the dose? Do you know how much the ibuprofen pills you buy are? They're hundreds of milligrams. I'm on 3 different prescriptions that are hundreds of milligrams for a single tiny pill. A 10 milligram pill is nothing. That is, I can't stress enough how little that is and how easy it is to make 10 milligrams of a thing. In lab, I'm making 10 milligrams as like the pilot studies of just like proving that we can do it at all. I'm getting enough that these people need total.
Here's the problem with that. Such little pill. You lose them.
You could lose them.
Pour them out. Yeah. Floats into the air.
You'd have to make sure that you'd weigh them down in the rest of the formulation so that they don't blow away.
Are you talking about fillers?
Yes. You'd have to stuff.
Is that allowed? I mean, you're going to get teased either way because your balls are going to get over.
Yeah, I figured you'd stuff anyway just because of the ball strain. Yeah. But I think it might be necessary here.
Okay.
So figure 4 is just that. It's the curves of how much it takes until your pregnancy rate for all of your monkeys are 0. You no longer have monkeys getting pregnant.
I never did to begin with.
I mean, then your figures would look the exact same.
Oh, I thought you were going to say my figure would be beautiful.
No.
I guess that could never happen.
Not without pregnant monkeys. Up to this point, this is where you have the developmental biology. This is when you have the animal studies. This is when you are being a med chemist. Now is you're actually... So the next question is an academic question that isn't necessary for drug studies, but is the question I would have asked first, which is, what the hell is the thing doing? Like, it's neat. Oh yeah, it bends sperm. That's great. Why? How? How do you know it's not hormonal? How do you know, like, what are any of the reasons you know this is a good idea? The moment you get something like monkey sperm count goes down, mouse sperm count doesn't go down, is when I would point out, you don't know what the hell the thing's doing. Otherwise, you would be able to explain to me why that is. So, that's when the next set of experiments come in. Now, we have drugs on the market that we don't know what they do. And we've been using them for decades. We just don't know what it acts on. With pharmaceuticals, it's really result-based. As long as you aren't killing people and you're curing the things you need to cure, it doesn't really matter what it does. But that's a hard sell to the public, that you don't know what the pills do. Figure 5 is they do 3 different experiments.
We got globins.
To say, oh, we got so many globins.
We got globins.
They do 3 experiments to tell them, what does the drug interact with? And they do 3 experiments that are very different kinds of experiments that all should tell them the same thing. And when I initially read it, I said, why are they doing so many experiments? Tell them the same thing. And then I remember that I don't generally do experiments for the purpose of putting something inside a human.
You would never put anything inside a human.
I would refuse. It's gross. But
Jesus says no.
They're trying to put forth a medication. And so they want to do multiple orthogonal studies telling them the same thing. And that's a good idea. And my confusion was purely because apparently I don't know how to not kill people. Because I would have been like, oh yeah, figure 5A. That tells them everything they need to know. But that's not true. There's figure 5A through H. And the 3 experiments they do is essentially, very quickly, if I take all the proteins in your cell and I chop them up with protein choppers, the things that are bound to the drug are protected because they're shielded from the choppers, right?
I think it's a little too ornate to chop them up with helicopters. You know they're tiny, right?
Do you not know about tiny helicopters? I'm sorry, are you the scientist here? You know all the sizes of all the helicopters in the whole world.
I'm a helicopter pilot. I'm a tiny helicopter pilot. That is my profession.
You're a tiny helicopter pilot, or you're a tiny helicopter pilot?
I dual majored.
Well, that's not really a layperson then. I was misled.
Let's get lay.
Let's get lay. So the drug would shield the protein. You chop everything up and then you see what was left, and the things that are left are primarily the things that the drug shielded. That's experiment 1.
Would it be good to say what a protein is?
Sure. So a protein is just a big molecule that your cell makes to do a job. Proteins can bind things, they can unbind things, they can modify things, they can cut things up, they can build stuff. They're nature's little robots. They just do things.
So what is this protein?
So they find a couple proteins that get shielded by the drug, and they're kind of all over the place. That's why they'd have to do 3 experiments, because they see a couple things didn't get chewed up by their protein cutting. But you can't assume the drug interacts with all of them, because maybe something just survived the chopping. That's fine. So you need different experiments. And they have one where they put the drug on a bunch of beads, and then they pass all of the proteins in the cell past the beads and see which gets caught. Because if you like binding to the drug, you'll get caught by the beads.
Just like friendship bracelet beads?
Yeah. I mean, they're agoros, but that doesn't really mean anything.
It doesn't mean anything to me.
So yeah, they're just beads. You basically put them on a little ball, and then you roll the ball through the liquid, and then you see what comes off on the ball. And that's the stuff that likes the drug.
Okay.
So we got what grabs the drug, what the drug shields, that's 2. And then they have one where they basically have a protein in mind called SPEM1, which is just involved in making sperm. So there was like a guess of like, maybe it interacts with this. And they make basically the bead experiment, but with SPEM1. And they see that the drug interacts with certain things and interacts with SPEM. And then they were like, okay, the drug interacts with this guy, gets grabbed by this guy, grabs the drug, interacts with the thing that we think would be the target. This is probably the thing that they found. And what they found was called junction plakoglobin / gamma-catenin.
Oh, we got the globins.
They got the globins.
Oh, covered in globins.
And the only thing any of that means, all that means, is that in figure 6, they draw this crappy little cartoon, which I think is
Jup?
A choice. Jup is the plakoglobin.
I don't know why they shortened it to Jup instead of globins.
Junction plakoglobin is Jup.
Yeah.
I don't know. What's SPEM mean? Like, no, this is biologists, okay?
Sperm is good because it's reminiscent of sperm.
It sounds like sperm. It kind of looks like sperm, the thing they drew. This is bad. Kurt, I mean, it's fine. It's not what I would have drawn, but it's fine.
What would you would have drawn?
I wouldn't have made them these weird shapes. Like, why is Triptonide that shape? It's like a weird sled.
What shape should Triptonide be?
Why does it need to be just make it a circle? Make any of these circles.
If it's all circles?
They've dumbed it down to being a cartoon with 4 colors. But then they tried to make fancy shapes for no reason? No. Either dumb it down enough that it's actually a dumb cartoon, or keep it technical enough that you're actually showing structures. They tried to do like half and half, and I find it very awkward.
As a single lady out here trying to get lay, this figure is probably the most confusing to me. I have no idea what this is trying to say.
Yeah, see, this is why.
It doesn't even list any globins.
It doesn't even say globe. Yeah, they did the abbreviations here, even though they didn't over here when they were describing the data. No, it's not the best figure, which is a shame, because as far as I'm concerned, this is probably one of the most important points in the entire paper. SPEM1 is in charge of parts of sperm morphology in the sperm generation in your body.
That was always my favorite Harry Potter class.
SPEM1?
Sperm morphology.
I'm just picturing Maggie Smith.
I'm always picturing
Dame Maggie Smith talking about sperm morphology.
God dang Maggie Smith.
Mm-hmm. That's what the Queen said.
A fine lady.
When she damed her. SPEM1's in charge of sperm morphology at some point. JUP is supposed to bind to SPEM1 and do something. Triptonide binds JUP in such a way that JUP cannot bind SPEM1. And they think this because of all of the other data talking about all of the things they said. And then they looked at mice when you knock out SPEM1, when you take that gene away and grow the mice and then look at that mouse's sperm, it looks exactly the same. So basically, Triptonide is turning off SPEM1. So if you know from this data that Triptonide is turning off SPEM1.
Well, it looks exactly the same as someone taking as mice.
As the mice on Triptonide.
Okay, not as just a normal mouse.
No.
Okay.
It looks exactly the same as what they were seeing with the mouse on Triptonide.
Okay, thank you.
So, if you have all of this data saying Triptonide binds JUP, but then this other data saying when you take Triptonide, it acts like it knocked away SPEM, that basically tells them Triptonide's job is to block JUP from binding SPEM. Because they don't have any evidence over here with the other 3 experiments I said. Triptonide isn't binding SPEM. They don't have evidence that that's happening.
Can you, for the love of God, refer to JUP as globins?
As globins. So, but there's so many different kinds of globins. This is just one.
Yeah, we gotta be covered in globins, but there's only one globin we're talking about here, so that's the globin.
This is the contextual globin.
Yeah. So the cunt globin, if you will.
So all of this comes down to essentially, we know you become infertile. We know they become.
They know.
They know they become infertile from deformed sperm. They know SPEM1 is in charge of sperm morphology. They know that if you take Triptonide, it looks an awful lot like you got rid of SPEM1. They know Triptonide doesn't interact with SPEM1 directly. They know it interacts with the globin.
Thank you.
Therefore, Triptonide is interrupting the globin's interaction with SPEM1 such that you can't form sperm properly. That's the end of the study. That is the bottom line. We know it works. We know it's reversible. We know it works and is reversible in primates. We know what it binds. We know why that binding affects the sperm morphology in the first place. This works, and when we made it synthetically and could administer it in a pure fashion, it was an effective drug. When we tried to purify it out from the herb, it was toxic because of these other things. They also did a couple experiments trying to figure out, like, making sure it's not toxic because it's an epoxide, which is a term people know from outside of chemistry because epoxies are things. And epoxides are dangerous and reactive and toxic.
And they adhere to things a lot?
The reason epoxies adhere to things is because an epoxide group in organic chemistry is, picture a triangle, and one of the points.
You know my brains don't do shapes.
Picture something
my brains, both of them.
3-sided with 3 points.
I got it.
One of those points is an oxygen.
Okay.
And the other 2 are carbons. This is a not stable structure. That's under what's known as steric strain a lot. The oxygen would like to react with anything else. It does not want to be kept in this triangle. It's difficult to be in this triangle.
It's toxic polyamory.
So it's exactly what it is. So the oxygen wants to break off and do anything else. So it's incredibly reactive and dangerous under the right conditions. The reasons...
just like any man.
Those epoxies that you use as adherents have violently reacted with the things around them and formed very, very stable bonds and don't want to go back to being epoxies. As is my understanding of how epoxies work. So when you if you have a drug with an epoxy group, you have to make sure it's not really, really reactive with stuff. And they make sure that didn't happen. They make sure it doesn't insert itself into DNA, because Triptonide, the chemotherapy drug, you have to make sure that it's, chemotherapy drugs kill cells. That's their point. They kill cancer cells. So they had to make sure that this wasn't chopping up your DNA or inserting itself or like stopping your cells from being able to replicate, or they had to make sure of all of those things. And they did. And they looked for a bunch of biomarkers to make sure it didn't happen. So they made sure that this is safe, and seems to be very, very safe. And I imagine from this data, it will go to clinical trials.
So this SPEM gene in mice, is it the same gene in monkeys?
So that's a great question. And they talk about, at the end of the article, they make some claim about sequence identities between So phylogenetic analyses have shown that mouse and monkey SPEM1 proteins share 69% amino acid sequence similarity, suggesting blah, 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 blah. The genes do the same thing, are in the same pathway, at least this pathway.
What do you mean by pathway?
The pathway of signal tells signal, eventually tells SPEM1, which somehow tells the sperm to not bend, apparently. So you have some sort of cascade of things interacting with each other, and SPEM1 is involved in the one that makes sure sperm is the right shape. So they know that it's doing the same thing with mice and monkeys, but they specifically mention something in that 30% that's not the same in the gene sequence is likely responsible for whatever it is that monkey SPEM1 does that causes monkey sperm count to go down and mouse sperm count to not go down. There's something about the protein that it's interacting with some other pathway, some other thing. Monkey SPEM1 does something else or something more or something slightly different. Now, the concept behind it being that 30% that's doing the difference, I would say, as someone who is trained in protein evolution, that is a mistake to make that deduction, because there could be something else in the monkey that interacts with the same spot that's present in mouse. Like if they were exactly the same SPEM1.
That's the hatred.
Monkey hatred?
Monkeys are filled with hatred and mice are not.
They are a wholesome creature. You could have the same exact SPEM1 protein in both mice and monkeys and still have a different effect from knocking them out, because monkeys could have a third thing interacting with SPEM that mice don't have, and that could be causing the difference. So the fact that they jump to the sequence identity question of, is this the same gene doing the same thing? I think your instinct to have that question was correct. I think their deduction from the answer is unsubstantiated. It's not wrong. I can't say it's wrong because they might be right.
So whatever in the monkey is going on that isn't going on in the mice that's changing things. Is that something they're pursuing? Do they want to isolate, figure out what that is? Is that something that you could figure out how to get around?
It depends on how much you would know about the developmental pathways in Cynomolgus? Whatever kind of monkey it was. Whatever that type of monkey is, you'd have to know about its developmental pathways for how it creates its sperm. Because it could be anything. Like, with other monkeys, they could do the same study in bonobos and not find that. It could be that monkey-specific thing. Who knows?
So it wouldn't be beneficial, really, to pursue that. They should just go to humans and see what happens there.
Probably, to be most time effective, yeah. Probably just go to humans and do the experiment. I had instructors in graduate school who would just be like, at the end of the day, you just have to do the experiment. Like, there's no matter what you do, eventually you're gonna have to put it in humans and see if it does something. So you might, as long as you don't think you have no reason to think it's dangerous. Sure, you might lower all the sperm count and you didn't want to, but then like, it's not like it killed them.
The sperm count goes back up after?
In the monkeys, yeah, when they stop. Yeah, the monkeys, it's apparently completely reversible. They go back to as if it never happened. And they had monkeys on this study lasted years. They had monkeys on this for years. And then go off, and then it was fine.
What group put this out? Was this a pharmaceutical company? Is it a school?
This was the Department of Physiology and Cell Biology in the University of Nevada, is a couple of them. Some reproductive and genetics lab in China, Family Planning Research Institute in China, Reno School of Medicine, the Biomedical Innovation Institute in California. It was multidisciplinary and across different schools, apparently parts of China, Nevada, and California.
What a beautiful collaboration.
Right? It's authorship. There's like 20 authors here. And that is, for something like a Nature's paper, that is pretty, not standard, but it's pretty frequent, because the only way you get into Nature and Science as journals is because you've done something that's a huge fucking deal. And the only reason, the only way you can be a huge deal is if you're pulling a lot of areas of expertise together all in one spot to study something important. If this is real, this tracks and scales and pans out the way it could, how many men of reproductive age don't want children but don't want a shot and don't want to wear condoms?
5. Probably
at least 5. And if it only takes 10 milligrams a day, you could take it with your multivitamin. You could put it in with a multivitamin. If it's only 10 milligrams, you could put out a men's 1-a-day multivitamin with this in it and just be like, this is the multi-day vitamin that also is a contraceptive. It would be really easy. It would take... No, I can't stress enough about how little 10 milligrams is as far as drugs go. It would be so easy.
Can you get into what the heck a globin is, and specifically what a junction placoglobin is?
I don't know what junction placoglobin is. I do know globins are... Globins! I'm pretty sure globins are just
like hemoglobin?
Yeah, they're just big bunches of a ball protein, just a big mass. They're globins. They're globins, and they bind stuff or get bound.
Like a supergiant of proteins.
Of proteins, yeah. I don't think there's anything special about junction placoglobin / gamma-catenin. I mean, it certainly sounds like it's an important thing. There are millions of pathway proteins that just kind of exist and bounce around and bind certain things through signaling pathways. So one of the problems with reading a study in not your field is someone could say any protein or drug or molecule or anything and not explain it because they assume you already know. So it could be that anyone reading about contraceptives or sperm production or reproduction,
they know these globins.
They're like, oh yeah, junction globin, of course.
This is globin like the back of my hands.
Just like if you said, so if this was my kind of study, and they just said the Michaelis-Menten parameters are blank, they wouldn't explain what that means.
Yeah.
They would just assume you know what a KM is, because why would you be reading this kind of study if you didn't know that, is the thought. And I've the studies that I'm an author on, that's the kind of, there's just so much you can explain. So it doesn't look like they say anything about the globin. And that might be because it's not really that important of a protein. It could just be one random signaling protein among hundreds. Or it could be a really important one, so important that they don't feel the need to explain what it is.
Would you take this, even if it ends up meaning that people are going to make fun of your balls for the rest of your life?
Bold of you to assume that they don't.
I guess I have heard tell that they're a little uneven, even more so than is socially acceptable.
You can't trust everything you read on the internet. No, they don't say anything. All they say is JUP, also called gamma-catenin. Given that catenins are the most common contaminants in mass spec-based proteomic studies, other proteins remain the more probable target. That's pretty funny, actually. They found it in their mass spectrometry data, and they're like, well, this is usually the contaminant, because these things are everywhere, so you find them in these experiments anyway. We had to do more experiments. And then it turned out to be that's actually the one that matters. So I think that even substantiates what I'm saying of just like, there's lots of globins. This one happens to interact with SPEM1.
Do you see yourself? Is this
right. I never answered the question. Yeah, sure. Why not? I don't mind or care, especially if it doesn't do anything. As far as the mouse study was concerned, it does 0 things other than make it so you don't have children. With the monkey thing, it's more concerning because there were more other unintended or unexpected effects. So I'd be curious to see in the clinical trials of like what happens. But hormonally, the monkeys were fine. Like you lower their sperm count, but the other charts are saying like their testosterone's fine, their FSH is fine. So you're not affecting their hormones, you're affecting like things like sperm count and maybe testes shape. So with that, I don't know if there's any lasting effects. So if it were hormonal, you could have all of the problems that is currently the pill problem, which is you have different changes in mood, you could have acne, you could have aches and pains. I do. You start screwing with the hormones of the body. You could really screw. Like hormones are the way your body talks to other parts of your body. So if the question was
and dance
and through dance, interpretive hormones and interpretive dance. If you ask me, would I take the pill in its current form right now? I would say no. It seems terrible. But I have the luxury of having external genitalia, so I could wear a condom. Whereas female condoms exist, but are more difficult than just...
I've heard they're a myth.
Unfurling of a piece of rubber, or whatever they're made out of. So
it's goat intestine.
You're not supposed to do that with goats. This pill? Yes. The pill? No.
Do you think that this could spread and replace the female pill?
So that's when you get back to where we started, which was there's the inherent politicism and the sexism and all of the onus is on the female because they give birth, that it's their job to be in charge of reproduction. I don't need to take it. Like there's there's reasons societally that this replacing the female pill is not a given. I think it should, because if one is hormonal and one is not, the one with basically no side effects should be the one that should be the prevalent form of birth control. But I don't know if that would happen, because my interest in the study ends at the science. My expectation of the society and what the drug, in however many years it will take, what that will do to the market of contraceptives, I don't know. I can't say. I'm a pessimist, so I would say no, I probably wouldn't replace it, because when do women catch a break? Like, but it could.
Sing it, sister.
It could, because I'm not enough of a pessimist to say everything will always be as sexist as it always has been, because empirically that's not true. So it could, especially if you do something like I was just saying, whereas if you market it like it's part of your daily vitamins, you bundle it up with your calcium and zinc for the day, like that sort of thing, it would be easy. It'd be so easy. Because it wouldn't even be like, oh, I have to take contraceptive, that's stupid. It would be just part of what you take. And the very first few lines of this talk about how overpopulation and unintended pregnancy are very real social and economic issues. They don't even go into like, wouldn't it be cool to not have babies? They're like, this is a real thing. And so far, society, we haven't come up with a male contraceptive that's super easy to use, or a female contraceptive that doesn't suck. So if we could get those, that would be great. Plus, would the church allow it? Would insurance companies care? Like, it's hard enough to get insurance companies to cover the current pill. So, like, there's a lot that could go into it. I think it should. I think it's really cool.
Well, is there a guarantee that this is going to be pharmaceutical and not like a supplement you get at GNC?
Nutraceutical would be the term.
A nutraceutical.
A nutraceutical, yeah. It depends on how they want to market it, and it depends on what the FDA would like to do about it. Because one of those, you need to go through very different pathways. I've recently learned. One of those, you need to go through very different pathways to get onto shelves. Because if you market it as a drug, there's a lot of red tape that you have to go through with the FDA. If you market it as a nutritional supplement, and it doesn't have any like adverse medical effects, so you could just say, yeah, just take it, it's whatever. Just like you could bundle up you could crush the root of the herb, the whatever herb they're talking about. You could crush the root and put it in a little capsule and sell that on the shelf, and you're like, what? It's a supplement. That would be fine. People do that. So you take this one molecule out from it, and it shows that there's not any adverse medical effects. It is very possible you could just sell it at CVS. This is a male contraceptive now, just like you could with condoms. But when things become an oral thing, and now all of a sudden you have to take it as a pill, people freak out because now it's a medicine, and medicine's scary. Or this could be in a box right next to condoms. That would depend on the company that decides it wants to make it.
So my final question is, what is the future of this science?
Of Triptonide? Specifically?
Potentially, or other things that occurred in these studies.
Well,
like where is this paper going?
It's in Nature, so it's far-reaching already. People, especially Nature Communications, it's very easy to disseminate that knowledge because it's not a 30-page study. Though there are 15 supplemental figures, so that's pretty normal for a science and nature paper. Science or nature paper. So it's easy to disseminate the knowledge, so the science that's actually going on in this study could be picked up and taken places. I'm sure there's an academic lab somewhere that will try to figure out what the globin and SPEM1 are really, really doing if that mechanism's not known. I'm sure there's an academic lab that will figure out exactly what pathway SPEM is working on that makes it so the monkeys have one thing and mice don't. I mean, like I already referenced, have years and years of training in protein evolution. My easy project for a grad student could be that, is there anything in that 30% difference in those spends that could indicate that they would have a difference? So from an academic standpoint, there are a lot of open, easy questions that you could make a proposal for and get funding for, because you'd be running with research that's coming out of a Nature paper, and that's like the magic cheat code for getting funding. From an industrial standpoint, I think anyone who wants to pick up Triptonide probably has a leg up on any competition, because this is out now, which means anyone else who's coming up with a reversible non-hormonal male contraceptive is now by definition playing catch up. Even if theirs is better, even if theirs is cheaper, even if you need less. This is the one that's currently on the books as being the best candidate. And therefore, this is the one that has the most credence. So if anyone wants to run with this, if there's any small Cambridge biotech startup that wants to say, well, we can make it, which seems to be what they're implying, is that they want a company to say, because they mention chemically synthesizing it and how that's an annoying thing, and how purifying it from the herb is also annoying and is often doesn't get rid of the toxins. So they're basically daring people in the discussion, go make this some other way where you can make a bunch of it really quickly. We made it this way and we weren't pleased with it. But we're a set of academic labs and like a health institute, which is probably where they did the monkey studies. So this could go really far. This could be a big deal. This could be the paper that's the cornerstone paper. That's the Nature paper that turned into the medication that was the blockbuster 10 years from now. It reads like it could be that, especially because it really seems like they covered their bases other than the things that you pointed out of, well, what's Spelman doing and what's Globin doing? But like I said, they might not have covered that base because the base is already covered. We just don't know about it because we're not reproductive researchers.
I'm a tiny helicopter.
I thought you were a tiny helicopter pilot.
I triple majored.
So I don't know how far it will go, but it could go really far. I think it's poised, and I didn't catch any complete BS from it. But it's not my field, so it could be BS.
Well, I would like to personally thank ninth author McSwiggan and all of their colleagues for this wonderful paper
There's a McSwiggan
of mice and monkeys.
Hayden McSwiggan.
And thank you, Dr. Jacob Wirth, for being a fantastic wingman and helping me personally get lay. Do you have any idea what our article for next week is going to be?
No, not at all.
Great. See you then.
I'm sure it'll be something.